Welcome to the deep dive. Today we're doing something a little different. We're taking a single, just a monumental source, a foundational chapter on the anatomy of the cerebral hemispheres, and we're going to try and boil it all down.
We are. Our mission here is to give you a complete visualized roadmap of the cerebrum.
Right.
We're going from the biggest, the surface features down to the microscopic wiring and then the huge white matter bundles that connect everything.
Think of this as the ultimate shortcut to the brain's blueprints. So let's start with the surface, because the brain isn't smooth at all, is it? It's intensely folded.
It is. And those ridges you see, those are called gyri. The valleys or grooves separating them are the sulci.
And those aren't just little creases.
Not at all. They're typically 1 to 3 centimeters deep. They're these deep extensions of the subarachnoid space, and they just dramatically increase the total surface area of the cortex.
And if a sulcus is particularly deep, we call it a fissure.
Exactly. And what's really useful, for getting your bearings, is knowing which of these are constant.
Because some of them are highly variable.
Right.
Right. I was surprised to learn that the little supplementary sulci can vary wildly from person to person.
Wildly. But the big players, the long primary sulci, so the lateral fissure, the central sulcus, the calcarine, the cingulate, those are reliable landmarks. They're on everyone's map.
So they're your core navigational points.
That's right.
And if you want a quick mental image of the brain's inner boundary, you just have to picture a continuous C-shaped ring on that medial surface.
Yes, that C shape. It's a hugely important anatomical feature. It's often called the great limbic lobe.
And it's formed by 2 continuous gyrus.
2, the cingulate gyrus arching over the top and the parahippocampal gyrus curving around below. That C shape is really your first hint at the deep circuitry for emotion and memory.
Okay, so that's the macro level map. Now let's zoom in way into the microarchitecture of the neocortex, the what anatomists call the isocortex.
Right, the fundamental 6-layered structure. And this is where we see how the work is really divided up at the cellular level. The cortex mainly uses 2 major types of neurons.
And their shape dictates their job.
Completely. The first, and by far the most abundant, are the pyramidal cells.
And they're called pyramidal because
Their cell body, the soma, is literally triangular. It's almost flask-shaped. Picture one single thick apical dendrite. That's the main input receiver going all the way up toward the surface, layer Y.
Okay.
And then it has multiple basal dendrites that spread out horizontally. They are the great excitatory neurons of the cortex.
So they're the ones telling other parts of the brain or the body to do something.
Precisely, to fire. And what's really interesting is that before the axon even leaves the gray matter, it shoots off this crucial recurrent branch.
What's that for?
It allows for local communication. It basically checks in with its neighbors before sending the signal way off into the white matter.
Yeah, and within this family of pyramidal cells, there are some
Yeah.
giants, right? Specialized for certain functions.
Oh, absolutely. The most famous have to be the Bet cells. You only find them in the main motor command center, the primary motor cortex, or BA4.
Oh, they're huge.
They can be 10 times larger than a common pyramidal cell, and they need to be, because their massive axons form the high-speed corticospinal and corticonuclear tracts. They're sending motor commands all the way down the spinal cord.
Wow. So if the pyramidal cells are the excitatory messengers, who handles the brakes?
That would be the other major category. The stellate cells, or as we call them, interneurons.
Okay.
These are local, primarily inhibitory neurons that use GABA, and they are incredibly diverse. Basket cells, chandelier cells, you name it, they're all about control.
And how do they control things?
They target very specific spots. So a basket cell will wrap around the cell body, the parasympathetic region, but a chandelier cell, it terminates right on the initial segment of the axon.
So it's like putting a powerful stop sign right at the launch point of the signal.
That is the perfect analogy for it.
Okay, so let's place these cells into that 6-layered architecture. How is work organized from layer to layer?
You can really simplify the 6 layers down to 3 basic rules of communication. So layer 4, the internal granular layer, that is the key receiving zone.
The post office?
The post office. If information, especially sensory info from the thalamus, is entering the cortex, it lands primarily in layer 4.
So that's the inbox. What about sending messages out?
That's where layers 3 and V are critical. Layer 3, the external pyramidal layer, is the main source of cortical output.
Meaning it talks to other parts of the cortex.
Exactly, to other areas in the same hemisphere or across to the opposite side.
Okay, so if layer 3 is talking to the rest of the brain, who's talking to the rest of the body?
That would be layer V, the internal pyramidal layer. This is our main descending motor output. It's where those giant BET cells live. Their axons are headed for the spinal cord.
What about layer 1, the
Mhm.
one right at the surface? Is that just empty space?
It looks sparse, but it is critically important. It's full of the apical input branches, the dendrites reaching up, but during development, it housed the Cajal-Retzius cells.
And they release reelin.
They release reelin, which is this glycoprotein that acts as a crucial stop sign, telling migrating neurons where to settle. If reelin fails, well, you get lissencephaly, the smooth brain condition. It shows you that the architecture is absolutely everything.
Input in 4, output to cortex in 3, output to the body in V's. It's a perfect functional roadmap. Let's shift to a highly integrated region, the limbic system.
Right, the center for memory and emotion. And the limbic system is less a single thing and more an interconnected assembly. Our source breaks it down into two key networks.
Okay, so first you have Yakovlev's circuit. That's the amygdala in the orbitofrontal cortex for emotion and reward.
Correct. And then you have the famous Papez circuit, which is centered on the hippocampus, and that is all about forming episodic memories.
Let's focus on the hippocampus because it's structured differently, isn't it?
It is. Unlike the six-layered neocortex, the hippocampus is archicortical. It only has 3 layers, and it's composed of the cornu-amonis, the CA regions, and the fascia dentata.
For anyone trying to really grasp how memory physically happens, we have to talk about the trisynaptic circuit. This is the information flow.
It is the anatomical basis of memory encoding. Think of it as a 3-stage relay race. It comes from the entorhinal cortex,
Okay,
and it enters the fascia dentata through something called the perforant path. That's stage 1.
got it. Where does the baton go next?
From the fascia dentata, it's relayed via these unique connections called mossy fibers to the CA3 region.
Stage 2.
Stage 2. And then finally, CA3 uses Schaefer collaterals to relay the information to CA1. That whole circuit entorhinal to dentata to CA3 to CA1 is what you need to learn new things.
Which explains why bilateral damage there is so devastating for memory.
It causes severe amnesia, absolutely.
That's an incredible visualization.
Right.
While we're on the topic of older cortex, let's quickly touch on the olfactory system, the paleocortex.
Right. Smell is unique. It's the only sense that bypasses the thalamus entirely. The olfactory nerves go through the crubriform plate straight to the olfactory bulb.
And the output from that bulb is really diverse. The lateral olfactory tract hits the primary olfactory cortex, sure, but it also sends signals straight to the amygdala.
Linking smell directly to emotion.
And to the entorhinal cortex.
Linking it to memory. That direct link is incredibly potent. And clinically, we now know that olfactory dysfunction is a major finding in conditions like anhedonia.
Let's move back to the 6-layered neocortex and look at functional mapping, starting with the frontal lobe and motor control.
We mentioned those BET cells live in the primary motor cortex BA4. What's amazing is that BA4 is largely intrasulcal.
Meaning it's tucked away deep inside the wall of the central sulcus.
Exactly. And it has the famous motor homunculus, that topographical map of the body.
Which is a very distorted map of ourselves, isn't it?
It is. The hand and the face are represented by these enormous areas, which just reflects the massive amount of cortex you need for fine-tuned dexterity and movement.
And just below that, still in the frontal lobe, we find Broca's language area, BA44 and 45.
Yes, and this area is intensely specialized. BA44, the opercular part, is mainly for syntactic processes, the structure of language. It's actually dysgranular. Its layer 4 is sort of invaded by pyramidal cells.
Whereas BA45, the triangular part, is for semantics, the meaning of words, and it keeps its granular structure.
It does, and we know that the volume of the left BA44 tends to be consistently larger in most people, which correlates really strongly with left hemisphere language dominance.
So if we cross that central sulcus, we land in the sensory domain, the primary somatosensory cortex, SI.
Right, in the postcentral gyrus.
Right.
And areas within it, like area 3B, are classified as coniocortical.
What does coniocortical mean for someone listening?
Think of it as a cortex built primarily for receiving, not sending. It has extremely dense cell packing, and most importantly, a really thick and pronounced layer 4, the main receiving layer.
Perfect for sensory input. And the primary visual cortex, V1, shares that designation, but it has a very distinct landmark.
It does. V1 is unique because it contains the stria of Gennari. It's a distinct, heavily myelinated band that's actually visible to the naked eye right within layer IVB.
And beyond V1, Visual processing just explodes into specializations. VM for color, V5 for motion.
And that specialization is so absolute. Damage to V4 causes achromatopsia, you can't see color. Damage to V5 causes achinotopsia, you can't see movement, just a series of still frames.
Incredible. And that continues down into the temporal lobe, along the fusiform gyrus.
It does. The midfusiform sulcus actually points to these specialized zones for object recognition, specific spots for face perception, and even areas for reading, like the visual word form area.
That functional density leads us to the insula, which is sort of hidden away in the lateral fissure.
The insula is fascinating. It's split by the central insular sulcus into two functional regions. The front part, the anterior zone, is a granular. It's linked to limbic and olfactory functions, emotion, empathy, that visceral feeling.
And the back part.
The posterior zone is granular. It's all about data processing, somatosensory and auditory functions.
And this separation is reflected in what you called a multi-receptor fingerprint.
Exactly. Different cortical areas have unique concentrations of receptors for every neurotransmitter. For example, BA4, the motor cortex, has the lowest overall concentration of most receptors.
Why would the motor cortex be so empty of receptors?
Well, one theory is that it's so highly myelinated for fast output that it actually prevents a high density of synapses from forming.
I see. And on the flip side?
Primary sensory areas have really high densities of inhibitory receptors like GABA. The thinking is that this intense inhibition is needed to improve the signal-to-noise ratio, sort of quiet things down so you can process all that incoming sensory data.
Okay, that makes sense. We've gone from the macro to the molecular. Now, if layers 3 and V are the senders, let's look at the infrastructure, the white matter, the cables.
Right. The long myelinated axons. They fall into 3 classes. Association fibers connecting areas in the same hemisphere, commissural fibers connecting the 2 hemispheres.
Like the corpus callosum?
The corpus callosum is the biggest one, yes. And finally, projection fibers connecting the cortex down to the body.
Let's focus on a few key association tracks, starting with the main output of the hippocampus, the fornix.
The fornix runs in this massive C shape right under the corpus callosum. It's the memory highway. Anteriorly, it splits into columns that project to the hypothalamus and, more famously, the mammillary bodies, which completes that trapeze circuit we mentioned.
Then there's the cingulum.
The cingulum runs inside those C-shaped gyri we started with. The dorsal part connects areas of the default mode network, the network that's active when you're just thinking or remembering. The ventral part is more for spatial orientation and navigation.
And finally, that hook-shaped bundle, the uncinate fasciculus.
A critical tract for higher cognition. It connects the anterior temporal lobe, including the amygdala, directly to the orbitofrontal region. It's vital for things like lexical retrieval and social cognition. Damage here can cause severe deficits in social behavior.
So we've covered the internal wiring. Where does that major projection output from layer V actually go?
All those massive descending fibers from the frontal and parietal lobes converge. They form this spectacular fan shape called the corona radiata, and then they funnel down tightly through the internal capsule to reach the thalamus, brainstem, and the spinal cord.
Wow. We has really traversed the anatomical foundations of this cerebrum today.
And we have, from the macro-level structure of gyri and sulci to the micro-level specialization in the 6 layers and the powerful white matter networks that tie it all together.
It just gives you such a phenomenal context for understanding the sheer functional density of every single millimeter of the cortex.
And to connect these details back to, our human specialization, remember those giant BET cells we talked about in layer V?
Yes.
Well, the key feature that really separates the human motor system is the strength of what we call direct corticomotoneuronal projections. This means the cortex connects directly to spinal motor neurons, bypassing interneurons. This highly developed direct control is believed to correlate directly with our dexterity, our capacity for skilled control of the hand.
So our microscopic anatomy dictates our ability to use tools.
Exactly. And that is something fascinating for you to consider the next time you use your hands for a complex task like writing or playing an instrument.
That is the perfect thought to end on. Thank you for joining us on this very detailed deep dive. From the Last Minute Lecture team, we hope you feel thoroughly informed and maybe a little less intimidated by the complexity of neuroanatomy.